fruitful and useful. First of all, I would like to introduce myself. My name is Aviva Blechman-Peretz. I am the Flow Cytometry Service Team Leader, and I'm also an application specialist on several additional instruments, and I'm part of the scientific support team here in Almog Diagnostic. Before we start, I would like to draw your attention, although I'm sure you have all noticed, to our new logo, which we have launched recently. So new logo, new year. This is also a time to tell you Happy New Year. Hope we will have healthy and less crazy year. So let's get started. So today, in order to understand the basic features of flow cytometry, we will briefly go over the basic components of the classical flow cytometer. This is actually a preface to our main talk that will focus on experiment design and setup. We will talk about panel design, antibody panel design. We will talk about the controls needed and all the points that are crucial to take into account when you plan your flow experiments. At the end of our talk, we will show you one of the coolest innovations in flow world today that we are very proud to be a part of. And of course, in the end, you will have time to ask questions. Please feel free to ask questions, to type in your questions in the Q&A screen. And we, of course, will answer all your questions in the end of our talk. So now to the basic part. So let's talk about the basic components of flow cytometer. As you can see here, we have this fluidics. We have the fluidics that moves the sample. And this is actually a very narrow stream of sheath where the cells are organized in a narrow line in order to get them one by one near the interrogation point, where the laser actually hits the cell. So this very narrow stream is achieved by laminar flow. And this is very important to get accurate data, to get the cells passing through the laser line one by one. So after the fluidics, we have the laser source. This is actually the light source to scatter and fluorescent signals. So after the cells meets the laser beam, the emitted light is directed through the optics to the fluorescent detectors. So the optics, these are actually the mirrors, the filters that we will talk about them in a few minutes, and also the lenses. They are actually responsible for directing the light to the correct fluorescent detectors here. So after the photons enter the detectors, the photons are converted to electrons. The signal actually exits the detectors as an electric current, and in this point, the signal enters the electronic system. The electronic system is not shown in this slide, but in electronic systems, the current travels to amplifier, and the signal is amplified and converted to a voltage pulse. And this voltage pulse is converted to digital number. This is processed by the computer. So in the computer, you will have the software that you can use in order to analyze your flow data. There are many very good softwares in the market. I'm sure you have heard about FlowJo, FCS Express. There are many other very good flow cytometry software that can be used to analyze your data. So before we will move on, I would like to explain about the filters actually. This is very important to understand the filter configuration when you build your antibody panel for flow. So there are mainly three types of filters: long pass filters, short pass filters, and band pass filters. The long pass filters here in this example, it's long pass 750. it means that wavelengths above 750 nanometers pass through, are allowed to pass through into the detector, and everything under 750 is reflected. Short pass filters are very similar, but the opposite, meaning that short pass 500 means that all the wavelengths under 500 nanometers are allowed to pass through into the detector, and all the higher wavelengths are reflected. And the most common filter in use in flow cytometry is the bandpass filter. Bandpass filter is actually a specific range of wavelengths, meaning in this example, bandpass 525/50, it means that all the wavelengths between 500 and 550 nanometers are allowed to pass through into the detector, and all the others are reflected. So actually this filter, this bandpass filter, is a common FITS filter. So after understanding that, I think we can go on and start to talk about the experiment design and setup. These are actually the 6 critical points that need to be taken into account when planning a flow experiment. I would say also from my personal experiments as a flow cytometry service team leader here in Almog, these are the main 6 points that I always need to address when I'm planning a new experiment. So we will talk about each of the points in detail in our presentation today. So you have to know the optical configuration of your flow cytometer. You have to know the characterizations of your cells, and you have to define the markers for phenotyping. You have to design the antibody panel. You need to take into account the antigen density on the cell surface. You need to take into account the fluorochrome intensity and the characteristics of the fluorochromes, because there are some fluorochromes that are not very stable after fixation. So all these things need to be taken into account. Another thing that you always need to think about is the unspecific staining, because in all biological experiments and biological systems, unspecific staining can occur. And we have to address it and do everything we can in order to minimize it. If it's the gate out of dead cells from your experiments, if it's the red blood cell lysis that needs to be done, and of course the compensation requirements and correct compensation is very important also in order to get accurate data. Controls that are needed for the setup of the instrument, meaning compensation and the voltage setup, the internal controls, the negative controls, we will also talk about them. And last but not least, defining the populations of interest, meaning the gating strategy. When you want to see a specific cell population, you need to know what would be the most correct and accurate gating strategy in order to accurately look at your data and get good statistics out of it. So the first thing we will talk about is the optical configuration of your flow cytometer. Also, we here in Almog, when we build panels for our customer, this is actually the first question: which flow cytometer you have, which lasers are available, and which detector bands are available. Because this is something we need to know in order to choose the right fluorochromes for your panel. So here is an example. This is a Milton, is a flow cytometer, Max Quant FL16, and it has 14 color options and 3 lasers. So we have the violet laser, the blue laser, and the red laser. And the detector bands that are available for violet laser, we have 5 detector bands. There are many fluorochromes that can be used here in these detector bands. For example, Pacific Blue, Vio Blue, DAPI. We have the Vio Green. We have the Brilliant Violet eyes, BD's Brilliant Violet eyes. So these are under the violet laser. Under the blue laser, we have 6 fluorescent channels, FITS channel, PE channel, and all the tandem dye channels that can be combined with all of our regular channels. and the red laser channels that are free on this particular flow cytometer. There are free detectors on the red laser channel for APC, for Alexa for 700 and APC size 7. So after knowing that, after understanding the capabilities of our flow cytometer, we need to discuss and know our cells characteristics. We have to understand if our cells express Fc receptors. If we are working with blood cells, we have monocytes, B cells, granulocytes that express Fc receptors. So in this case, we should consider using FCR blocking reagent. Another thing to consider is autofluorescence. Dead cells, let's say, can have very high autofluorescence, and it can alter our results afterwards. We can just get false positive results because of the dead cells. So it's very, very important to gate out the dead cells in every flow experiment. We will also talk about it in a few slides. And also important is to know the origin of the cells. Cells that come from organs, inner organs like lungs and kidney, they tend to have high autofluorescence. Tumor cell lines, senescent cells, all those have higher autofluorescence, which also needs to be addressed. So in case of cells that have a higher autofluorescence, we can always choose channels that do not show this higher autofluorescence, if it's possible. If it's not possible and we need to occupy all the channels of our flow cytometer, we need to choose brighter fluorochromes, bright fluorochromes, in order to distinguish well between the autofluorescent and the positive signal. Intracellular or extracellular antigen markers, this is also very important to know before. So if you are staining intracellular or intranuclear markers, you need to choose a proper fixation and permeabilization buffer set. Also, all your controls, your compensation controls, and all other controls need to be treated in the same way, exactly the same way, exactly with the same fixation permeabilization buffer set. Red blood cell lysis, many times, when we are running whole blood, it's sure that we have to use a red blood cell lysis solution. And I would also recommend to add a CD45 antibody in order to gate on white blood cells and get good statistics out of the data. In case of traces of red blood cells, let's say from spleen, mouse spleen samples, for example, we do not have to perform a red blood cell lysis, but we do recommend to add a CD45 antibody in order to get, again, good statistics of your cell populations out of white blood cells. And the last one is we have to address also this thing, the issue of the tendency of the cells to create doublets and triplets. As we said in our first slide, it is very important to get the cells pass through the laser beam in a narrow stream one by one. So in case we get sticky samples, sticky cells that tend to stick together, there could be two or more cells that pass through the laser beam, and in this way we get actually reduced accuracy. We get false positive signals. Of course, we can have also clogging issues that need to be considered. So if you do work with cells that tend to clump together, you should always consider adding EDTA to the FACS buffer. So now let's talk about defining the markers for phenotyping. There are really many tools today that can assist you. One of the great tools that Miltenyi has developed is a scientific knowledge center. You can reach this center by entering Miltenyi's website. This is just an example. I will show you how it works, but of course you have many other knowledge centers in the internet era and many informational platforms that you can use. So in Miltenyi's handbooks, actually you can find information according to the cell type. Let's see that we are interested. For example, look at pan T cells. Let's talk about T cells and see what kind of information we can get from Miltenyi's website. So of course we can get information about the cell subsets. We can get information about sample preparation and about separation of specific subpopulations. But what interests us today is the characterization of T cells by flow cytometry. So here you can get a nice table summarizing some of the T cell panels that are recommended. For NKT cells, you have this recommended panel for analyzing exhaustion of T cells, let's say in case of designing CAR T cells. So this is the panel that is also recommended to be analyzed. And of course, you have many other panels. You have panels for Tregs that you can find in this web page. So we really do recommend you to use this knowledge center. But of course, you are always welcome to contact us in Almog technical support team. It's me, Aviva, and Galia, my colleague. We are always willing to help you. You are always welcome to turn to our wonderful salespersons and product specialists, Ronit in her area of biotech companies, and we have Heli and we have Gal. So whatever is convenient for you, we are always happy to assist. And the last one is the literature. You can find lots of information in publications today in the literature, and it is really quite easy to get information about which markers to use for phenotyping of the cells of interest. So now we will move on to designing the antibody panel. So the two things that we need to consider when we design the antibody panel is the antigen density and the fluorochrom intensity. So antigen density means level of expression of the antigens on the cell surface, or in case of intracellular antigen, the expression levels of those inside of the cell or inside of the nucleus. So this is something that we need to consider. And of course, the abundance of the cell population. Let's see, there are actually 3 categories that are in use today. There are the primary expressors that express the antigens in high concentration, high density on their cell surface. And these are CD3, CD4, CD8, it's of course CD45. We have the secondary expressors. These are intermediate densities, and sometimes they have smeary markers. They have low, high, and medium expression levels. These are, for example, CD127. This is actually a Treg, a marker that we use for Treg phenotyping. And we have a territory markers that are low density markers. For example, CD25, it's a smeary and quite a low density marker. It is not expressed many times on the cell surface. So these are things that need to be considered when we go and build our antibody panels. And the second thing is the fluorochrom intensity. So fluorochromes are not equally bright, and also background has to be taken into account. So scientists have developed this staining index calculation. In very, very simple words, the staining index is actually the resolution, a value for the resolution between the negative and the positive peak. So this is the distance between the negative and positive peaks, and there are fluorochromes like PE size 7, PE Vios 770, or Viobright 550 that have very high staining index, meaning that they are very bright dyes. And on the other hand, there are the dimmer dyes like PerCP, but their staining index is much lower. So in order to make it a little more simple, scientists have agreed on a ranking of those staining index in a scale of 1 to 5. When 5 is the brightest fluorochrome and 1 is the dimmest fluorochrome. So this ranking system is actually in use in many companies where you can purchase antibodies. You can see this ranking of on scale of 1 to 5. So this is something that we also address when we build our antibody panels. Again, here, it is very important to show you that actually the level of antigen expression, the lower the level of antigen expression, the brighter the fluorochrome that you will have to choose for this specific antigen. Okay, and this is also, I would like to emphasize that you have to take into account the source of your cells. If you're working with cells that come from enzymatically dissociated tissues, you should always take into account the fact that the enzymes can cleave off your antigens from the cell surface. If it's cleaved off, absolutely cleaved off, you cannot do anything. But in case it's a partial cleavage, you can, of course, titrate the enzyme concentration. And you should know that you should choose a bright fluorochrome for the antigen that is partially cleaved off from your cell surface. So this is also something that needs to be considered. So now we have come to the part of designing the antibody panel. I will enter Miltenyi's website just as for example, to show you how the antibody panel builders work. There are many platforms available today. There are, let's say, Fluorofinder. Fluorofinder summarizes most of the antibodies offered by most of the antibody producing companies. So you can, of course, use Fluorofinder platform. But here, for example, I will show you Miltenyi's panel builder. So if you enter Miltenyi's website, you will get to the panel builder that looks like this, and you can type in your flow cytometer. I'm sorry, a little technical issue. And so you can type in. I'm sorry, I'm very sorry, it's not seen, but I will show you here. You can actually, you get this screen, there you can select the markers of interest. Let's say you can type in CD3, CD4, CD8, or of course many other antigens that are of interest. But one step before, you can type in your instrument you are using. And then you type in the instrument you are using, you are immediately getting all the available channels, and the panel builder will offer you only the dyes that can be used on your filter, on your flow cytometer. Okay. So in this case, I chose Miltenyi's Max Quantana Laser FL16. We have 14 channels, and we get all these available dyes that can be chosen for for the antibodies. So the next step will be you will get this table of all the antigens and the antibodies, actually CD3, CD4, CD8, and you will get a list of fluorochromes available. And as you can see, you have this ranking system. Here, BioBlue is ranked as 4. You have this ranking seen here, and FITS has 2. FITS is actually a dimmer fluorochrome. So here you can choose fluorochromes for each of the antigens. Again, you need to bear in mind the rules we talked about before. If these are primary expressors like all these 3 here, or there are secondary or tertiary expressors. So for CD3, CD8, and the CD4 and CD8, you can choose dimmer dyes like FITS, like Percipi, like also VioGreen and VioBlue. And of course, for your dimmer expressors, your lower expressors, you can choose PE Vio 770, this is PE Vio size 7, or ABC Vio 770. You have many options here. After you continue, you will get this nice table summarizing all the antibodies you have chosen. And you will get the catalog numbers, and you can email it to yourself, you can print it. If you email it to yourself, you can send it to us, and we'll send you a price quote. And we can, of course, if you need to consult, we are always happy to assist. And we can also assist you with the panel building itself. You can call us, we are always happy to do it. So now we will talk about the issues that need to be considered also, like the unspecific binding. We need to reduce unspecific binding. And in addition to binding to receptors of interests, the antibodies may also bind to other non-specific sites. There are a couple of unspecific bindings that can occur. One of them is binding of the antibodies to the cell surface through the Fc receptor. So in this case, we would recommend you to block the Fc receptors with the correct FCR blocking reagent. We can also get a non-specific binding through the fluorochrome. In this case, tandem dyes have higher tendency to unspecifically bind a specific subtype of Fc receptors on a subtype of monocytes. So these tandem dyes, this phenomenon can eliminate it by tandem signal enhancer. I'm sorry. The tandem signal enhancer is a FCR blocking reagent, which will specifically block also the special Fc receptors that bind the tandem dyes. So when you choose to use tandem signal enhancer, you do not need to use any other FCR blocking reagent because it blocks all the Fc receptors. It is very important to use accurately titered antibodies in order to eliminate any unspecific staining, any overstaining or understaining also. And of course, again, discriminate the dead cells in your samples. So accurately titered antibodies, there are many antibodies in the market that need to be titered before you use them for your flow experiment. You need to perform antibody dilution experiment that you find the correct antibody titer in order to get the best resolution between your negative and your positive peak. Okay, so this is something that you need to do really before you perform your actual experiment with your cells. There are many antibodies in the market that are already titered and calibrated, like Milteney's antibodies. You do not have to perform any additional calibration, any additional titration. You just add 2 microliters of antibody until up to 1 million cells. You incubate your cells for 10 minutes and wash your cells, and you can analyze them on the flow cytometer. This is very new and very innovative antibodies from Milteney Biotech. These are recombinantly generated antibodies. And what is very special and unique for these antibodies, first of all, they are all human IgG1 isotype, from a human IgG1 isotype, and they all have mutated Fc region, meaning that these antibodies, these rare clones, do not bind Fc receptors unspecifically. So you do not have to perform any blocking of your Fc receptors, of the Fc receptors. And one additional thing is that you need only one universal IgG1 isotype control that is conjugated to the dye of interest. And of course, they are pre-titered. You do not need to perform any calibrations for the amount of antibody. They have short incubation times and they are QC controlled. So Milteney, you can find those antibodies also in Milteney's panel builder, in Milteney's website, and we highly recommend them to our customers. So the next step that need to be done is we have to think about the dead cells, because we need to gate out the dead cells, and dead cell exclusion is extremely important, and it needs to be done in every antibody panel. Actually, we call them ghosts. The dead cells are like ghosts. They can give you unspecific signals. They can give you false positive signals. So it is very important to gate them out. There are actually 2 groups of dyes. There are dyes that are suited for running live cells. These are dyes that are based on DNA labeling. They are DNA labeling dyes. I'm sure you all know propydiumyodide, 7-AAD, DAPI. There are many, many others that can be used. And there are, when running fixed cells, there is a group of dyes that are amine reactive dyes. For example, ghost dyes, viability dyes, zombie dyes, they all bind the amine groups of the protein. So in case of dead cells, the dye managed to enter the cells and also stain all the proteins inside the cell and nucleus. And in case of live cells, this dye will only bind the amine groups on the cell surface. So in this then using those amine reactive dyes or also the DNA labeling-based dyes, you will get a distinct positive signal for the dead cells, and in this way you can gate on the negative population, which are the live cells. Okay, so now we will talk about compensation. Compensation is also something that we need to perform with every new antibody panel, or with every new antibody panel that we have. Wishful thinking is that every dye has its unique emission spectra that is captured by its unique filter, but actually in the real world, it is not, it doesn't work like this. In the real world, what we see is that dyes have overlapping emission spectra. Here is a fits in filter that is designated for fits, and this filter here is designated for PE. And as you can see, that the fits has a small emission portion of its emission in the PE channel. And this spillover of fits into PE channel needs to be subtracted from the PE channel. And this is true also for PE that emits partially into fits channel. So this is called compensation. We recommend to perform automatic compensation. All the machines today have this option, all the softwares today have this option of performing automated compensation. So then we are looking at uncompensated data. Let's say for fits single stain, we can see that in addition to a signal in the fits channel, we also see some portion of the signal in the PE channel, and this needs to be fixed by compensation. It means that the median fluorescence in PE channel for fits needs to be the same as the negative signal. So we just want to see this this imaginary straight line that strikes through the negative population and also the positive population. So we also have this scenario of overcompensation. I do want to tell you that in case you are using automated compensation, you really, most of the times, get very good data, and you do not need to touch or need to fix anything. You do not see these scenarios of overcompensation or uncompensated data. So there are 3 basic rules that need to be addressed. These basic rules will actually assist us to get the most accurate data for the compensation. We need the compensation controls to be stained with one color only. We call it single stain, and they need to be as bright as possible, meaning that as bright as your signal for your cells or brighter, but not less than that. You should use the same fluorochrome, meaning that if your cells express GFP, you cannot use fits for compensation because it's not the same emission spectra. It's not the same characteristics of fluorochrome. So this is something that you always need to bear in mind. Positive and negative populations, because you need the median to compare the median, or the software will do it for you, you need a distinct positive and negative population, and they have to have the same level of autofluorescence, okay? So in this example here, I am showing you the compensation beads, actually. Instead of cells, we can use compensation beads. The compensation beads, they exchange our cells. Instead of cells, we use compensation beads that bind our antibodies. And in this case, we see distinct negative signal and distinct positive signals. Here we have for fits and PE, and here we have the compensated data. So one thing that needs to be considered also when performing compensation and performing multicolor panels, you should consider the technical limitations of the tandem dyes. So tandem dyes actually can break apart upon if they are not valid anymore. I mean, if the shelf life, we need to pay attention to the shelf life. So in case that the tandem dyes are stable and perform as expected, we have this example of PE size 7. Here the PE molecule is coated with size 7 molecules, and by FRET, the PE gets excited and transfers the photons to size 7, which is actually responsible for emitting fluorescence. And in this case here, some of the size 7 molecules have fallen off the PE molecules, and in this case, we will also get a signal from the PE molecule. So we will get signal for PE size 7 and PE, and we will get false positive signals, and this is something we cannot fix by compensation. So you should always work with valid lots, and the compensation is also specific for the lot, because there can be small variations between the conjugations of the tandem dyes. And of course, this is also the place to remind you that you can use the tandem signal enhancer that improves the binding specificity of tandem dyes by blocking the Fc receptors. So today we have really a very high range of fluorochromes offered by many companies. We have many tandem dyes that we can offer for analysis. Miltenyi have developed biodyes. They are high fluorescent, they have high fluorescence intensities, and they are designed to have lower spillover to neighboring channels. So there are many dyes. In case you are interested to get more information, we are happy to provide you with. Yeah. So now we will look at the controls needed for our experiment. This is also something that I have gathered here for my, also from my personal experience. We will always need unstained cells, the same cells that will be used in the experiment. This is important in order to address and estimate the autofluorescence of your cells. We will also need single stain controls with distinct positive and negative population. This is that we need those single stain controls for voltage setup and also for compensation setup. This is important to remind you that the single stain controls need to be treated exactly in the same way as your cells will be treated. So if you are using fixative and permeabilize your cells, the single stain controls should also be fixed and permeabilized. You need to do it in every new setup or also in a new batch of tandem dyes. If you have a new batch of tandem dyes, you should again run single stain controls in order to perform a new compensation. Isotype controls and fluorescent -1 controls. These two controls assist us in gating our positive populations. So isotype controls, actually they are to control the non-specific binding of antibodies. And the fluorescent -1 controls are very important also for accurate gating, because we can get spread of fluorescent signals even after we compensate our antibody panels. And fluorescent -1 controls assist us in eliminating this fluorescent spread. I can tell you that fluorescent -1, this is something that the flow world recommends to do in every multicolor experiment. This is something that we actually consider very carefully. When we are staining intracellular or intranuclear antigens, we always perform fluorescent -1. When performing staining extracellular antigens that are abundant antigens, and we sometimes leave the fluorescent -1 controls out. And of course, you will have to pay attention to your experimental controls, untreated cells in case of treatments, or normal cells in case of abnormal samples. So this is also something that you need to think about. So one moment before we will look at gating strategy, I do want to talk about doublet discrimination. As said in the beginning of our talk, cells can be at very close together and excited by the laser as a single event. And in this case, we do want to gate out the doublets, the cells that are stick together or are very close together, we want to gate them out in order not to get false positive signals from these cells. So the only important thing to remember here is that the gate out of doublets is done in linear scale. So in most experiments, forward scatter and side scatter, we use them in linear scale. So the doublet discrimination is done in linear scale. And we, for example, let's just look at this first example here. We have forward scatter area against forward scatter H, and we get our single cells, singlet cells here in a diagonal, and all the cells that fall out of this diagonal part into the area region, these are the cells actually that are doublets or triplets, and they should be gated out. There are many ways to do it. All the ways are good, all the ways are acceptable. You just choose whatever is convenient for you. So for last and not least, defining the gating strategy. After you design your antibody panel and you know which populations you want to see, you want carefully to design also the gating strategy in order to see how you get the most accurate data and statistics for your cells of interest. So here is an example for phenotyping of white blood cells. We can see that the singlets have been gated forward scatter against forward scatter area against forward scatter height. The doublets have been gated out here. After looking at our cells, our single cells, we are looking at viable cells. We are gating out the dead cells which are positive for 7-AAD in this case. So we draw this gate for our live cells. So now we are looking at our live cells, and we are looking at the morphology, forward scatter against side scatter. We want to gate out the debris, the small particles. Afterwards, we will look at our leukocytes that express CD45. In this case, you can see that all these cells that are negative to CD45, most probably these are red blood cells, traces of red blood cells, and it can be also platelets. So we are gating on our leukocytes. After that, we are going forward, and we are looking at monocytes. So we are looking at CD14. We get our monocyte population here, and the non-monocytes, of course, are negative to CD14. So 14. So if we want to define classical and intermediate and non-classical monocytes, we can look at this gate here and again look into CD14 against CD16. So here we get this separation of non-classical, intermediate, and classical monocytes. If we are looking at non-monocytes, all the cells that are negative for CD14, we can go on and look at B cells, neutrophils, eosinophils, also T cells, NKT cells, and we can also look at the CD8 and the CD4 and CD8 T cells. And of course, we can go on, and we can gather our statistical data in a table. And this is really, there are so many ways to do these things, but this is only one example. So actually, we have come into the end of our talk. I do want to show you one slide about the really exciting new innovation in the flow world. We call the next chapter of flow cytometry. This is actually the spectral flow cytometry. And instead of one detector, one fluorochrom, the new technology enables us full spectrum analysis, which gives us a unique fingerprint to every fluorochrom. So in this way, actually, this is what makes it possible to get up to 40 colors in one tube and in combinations that you could not imagine in your wildest dreams. So here is one example. For APC and Alexa Floor 647, these dyes cannot be analyzed together in the same tube in conventional flow cytometry. since their emission peaks are almost similar. But in spectral flow cytometry, we get this spectral signature for each dye. And when looking at these signatures, the main peak is, of course, in the same area, but the other, there are other, this is the specific signature for all the other lasers and detectors. And we can see here differences in the violet channel. We see a different peak here in violet channel for APC. We can see differences also in blue channels. So these distinct signatures enables us to combine lots of colors that cannot be combined in conventional flow cytometry. So here is a nice example for you. Yes, we can see an analysis of Alexa Floor, CD56 Alexa Floor against CD8 APC, and you can see a nicely distinguished positive signal for NKT cells, and this is really exciting for me. So now it's your turn. You can ask questions. Please feel free to ask questions. And before that, I just want to apologize in the part that I mentioned that you can contact us for any help in building an antibody panel or designing phenotyping markers. You can contact our salespersons as product specialists. And I accidentally left out Neta, our wonderful Neta. So Neta is also available to you. Like all of us, you can call us, you can send us emails, whatever is works for you. So now, if you have any questions, I will be glad to approach. Please feel free. One moment. Okay, one moment, I will ask. I will answer, sorry. Do you still need to use FC block in case you use rare antibodies? So the answer is no, but in case you are using tandem dyes, when the unspecific binding is through the tandem dye itself, it is, you still should consider using a tandem signal enhancer, because this unspecific binding is not through the FC, is not through the isotype of the antibody, but rather through the fluorochrome itself. How I choose correctly the voltage, how do I perform the voltage settings? So the voltage settings are done also with your cells, with your single stains. The best way to do it is to do it with your single stains. You should always take, be sure that the signal is inside the plot and doesn't reach the borders of the plot. I mean, you have to see a good positive signal in the middle of the plot. You should do set the voltage in this state that you get distinguish between the positive and negative signal, but still that the signal does not run out from the plot. Is there a method to increase intensity of compensation beads in cases that the samples give a stronger signal? One moment, and to increase intensity of in case the samples gives a stronger signal. So what you can do is you can add more antibody per the same amount of beads. Okay, so actually in order to get the full staining of the compensation beads, the antibody should be added in higher volume, higher concentration of antibody per beads than you use for your cells. If you use 2 microliters of antibody for your cell staining, you should use 4 microliters of antibody in order to get the full staining of the compensation beads. So in most cases in this way, you get a stronger signal than you get for your cells. Okay, another question. Do you still need to use compensation even with dyes from different lasers? The answer is yes, you still need to do it because there are dyes that, let's say, if you are using dyes in the violet laser and dyes in the red laser, you do not need to perform any compensation because these dyes do not they are not excited by the dyes that spill into violet laser channels are not excited by red laser. But it can happen that, let's say, PE molecule. PE is excited by the blue laser and also by the yellow laser. But as an example, the PE is excited by the blue laser and its detector is under the blue laser, but also PE can be excited by red laser. So you can see a small portion of PE also in the APC channel. So yes, you have to, there are dyes that get excited also by different lasers. So you need to address it. I can say like an overall rule that dyes that are excited by the violet light laser, like Pacific blue or Pacific orange, these dyes do not spill into the red laser channels, but there are some dyes in the blue laser region and also in the red laser region that need to be checked because they can also be excited by adjacent lasers. Okay, so if there are no additional questions, I would like to thank you. I would like to thank you for attending this webinar. I hope you find it useful, and in case you have any questions, you are welcome to contact us. Yes, and I would like to remind you that next week we'll have a webinar about the spectral flow cytometry and the new cytometer, Aurora and Northern Light cytometers, sorry, from Cytec Bio. And you are all welcome to join us. And I think it will be very, very interesting for you. So hope to see you all next week if you don't have any additional questions. I do want to put here this slide just for you in order to enable you to write down our emails in case you would like to consult us or assist you with phenotyping panels, antibody panels, anything, everything. So have a nice day and hope to see you next week.